Good morning. My name is Prey Mohanty, and I am a life science product specialist and product marketing manager at Benchling, based out of the San Francisco Bay Area. With me, I have Tom. Tom, can you go ahead and introduce yourself?
Hi there, yeah. So Tom McCrory. So I'm a scientific solution consultant here at Benchling, and I'm going to be taking you through a live product demo of our topic today.
Great. Thank you, Tom. So welcome to the second webinar in our early development webinar series. In the first webinar conducted on October 12th, we discussed the Benchling solution for bioprocess development. In case you missed it, you can view the recorded version of the webinar on our events page at benchling.com/events. Today, we will be discussing bioanalytical development. In today's webinar, we will briefly talk about some of the challenges and key considerations in doing bioanalytical development successfully. We will follow this up with a live demonstration and wrap up with an audience Q&A. Please type your questions into the Q&A window to the right. Notice I think there's a chat window and a Q&A window. Please make sure to type your questions in the Q&A window so that other people can vote on your questions as well. We will answer all your questions during the Q&A session towards the end of today's webinar. Let's start. Bioanalytical is an integral part of preclinical and clinical development. Anytime companies want to evaluate the pharmacology, pharmacokinetics, toxicology, or bioavailability of drug candidates in animals or humans, bioanalytical has a key role to play in the quantitative analysis of the drugs, metabolites, or related biomarkers in biological samples. Consequently, the data generated from bioanalytical development is an essential part of regulatory submissions such as INDs, NDAs, and BLAs. Bioanalytical development today has evolved significantly, driven primarily by innovation in the space. The pipeline of biopharma companies has become increasingly diverse. In addition to traditional molecules such as small molecules and monoclonal antibodies, newer molecule types such as RNA based therapies, bispecifics, and antibody drug conjugates continue to push the boundaries of bioanalytical analysis. The second source of innovation is the development and use of novel biomarkers and related assays throughout preclinical and clinical development. As companies target rare conditions and difficult to treat diseases, they are increasingly relying on biomarkers for getting early reads on safety and efficacy, for patient selection during clinical trials, and for primary and as primary and secondary endpoints during clinical trials. The final important source of innovation is a large CRO industry that serves the biopharma industry. In addition to keeping costs low, CROs often provide innovative and differentiated bioanalytical assays and services. Many also have partnerships with biopharma companies to develop proprietary assays and technology platforms. So, what does successful bioanalytical development look like? We believe that there are four themes to measure against. They are data integrity, collaboration, insights, and quality compliance. Before we dive deeper into each of these topics, let's take a quick poll to understand your view on this. A poll question will pop up shortly. Just wait for a few more seconds before we close out the poll. Okay, I think we can close out the poll, Josh. Okay, great. So looks like inefficient data collaboration, controls, and even data integrity. All those three seem to be the key challenges for this audience. Now let's look at each of those themes that I just presented. So bioanalytical teams contend with significant project load with condensed timelines as they support key GXP study readouts. Teams want to minimize user variability in processing and analysis of biological samples that often originate from expensive preclinical or clinical studies. Data integrity becomes extremely important against this backdrop in ensuring accuracy and reliability of bioanalytical data generated. The topic is so important that both the FDA and MHRA have guidance documents on this topic. A good digital solution should improve data integrity by standardizing data capture across analysts, enabling consistent SOP execution, and providing full traceability of methods and samples used in the bioanalysis. Bioanalytical development is highly collaborative in modern R&D organizations. There are specialized subteams or team members that manage incoming and outgoing biosamples, develop complex assay methods, execute sample analysis, provide compliance oversight, and author reports and regulatory documents. Depending on the organizational size and setup, any or all of these activities can be performed by specialized team members. In addition to this internal collaboration, most teams also have extensive collaborations with CROs providing bioanalytical services. A good digital solution should help manage this complex collaboration. It should enable easy context sharing between team members, facilitate role based collaboration, and provide data management tools to manage varying degrees of access depending on role. Generating insights can be tedious and time consuming in bioanalytical development. Teams are often running a complex panel of plate based assays and chromatographic techniques. Each of these assays have raw data coming out of analytical instruments, processed analyzed data created by analysts, and final versions of results with statistics and interpretations that need to be recorded or managed. Additionally, there is data at the process or operation level, such as resourcing and project tracking, that are performed with different tools. The data from these disparate sources are often siloed. This means that to perform any meaningful data analysis, scientists have to manually aggregate data before they can run advanced analytics or draw any conclusions. An ideal digital solution should solve these data management challenges and provide timely decision support at the experiment, process, and operation levels all in one solution. Enabling compliance requirements is integral to supporting bioanalytical in early or late development. Every aspect of bioanalysis, starting from the biological samples to the method documentation, sample preparation and analysis, and results reporting, has to be traceable. This traceability facilitates compliance oversight and reduces regulatory risk. A digital solution must provide the necessary tools for enabling compliance with full traceability across samples, methods, experiments, and results. To truly meet the varied needs of bioanalytical development, which spans hypothesis-driven research through procedure-driven execution, a modern digital solution should combine the capabilities historically served by disparate ELN LIMS and LES software into one unified R&D solution. Modern electronic lab notebooks should not only document experiments and methods, but should also provide sample and resource management. LIMS and LES should have experimental context and biological understanding in addition to core sample traceability. Although processes need to be locked down for compliance reasons, solutions need configurability that adapts to varying program needs. Let's take another quick poll here. The poll should pop up momentarily. So the poll reads: What type of R&D work do you or your team perform more often? Is it exploratory research, controlled development, lockdown execution? And then you have some options combining those three options. So both one and two, two and three, one and three, and all three. We'll just wait for a few seconds for the votes to come in. Great. So looks like both exploratory research and controlled development, the combination of the two seems to be the most popular choice among this audience. And there's also a few doing all three. Benchling offers a unified suite of applications built on a trusted cloud platform that addresses the needs of R&D teams across workflows that are experimentally driven, sample driven, and procedurally driven. Benchling supports the distinct needs of bioanalytical development by supporting exploratory hypothesis driven research, control protocol driven development, and lockdown procedure driven execution, all on the same platform. So, going back to the poll of the audience, since most of you were interested in about two or three of the ways of doing R&D, so Benchling is suited for doing all three in one platform. In supporting GXP studies, bioanalytical teams require more capabilities for increased regulatory compliance, including for developing methods per guidelines, executing assays in controlled experimental setups, and enforcing quality compliance. Bioanalytical teams that work alongside preclinical teams such as in vivo pharmacology, safety, and tolerability must collaborate on sample intake, processing, and reporting back results that require full traceability. Being able to easily receive and assign tasks makes this back and forth work efficient. We know that teams work with large complex data sets. These can be aggregated by searching within Benchling Insights. With a line of sight to regulatory submissions, the bioanalytical solution provides end to end traceability for sample lineage and experimental context. We also found customers reported a 150% increase in ease of finding data and metadata by working within Benchling. With that, I will hand over to Tom to walk us through the live demo. Tom, it's all yours.
About bioanalytical development, it really spans a lot of different key deliverables. I have those here up on the slide, that kind of an overview of that workflow, but you'll see it's starting with method development and ending with validation at the end of that stage. And then once these methods are validated, the bioanalytical team will perform these validated assays on samples that are received from maybe like a preclinical in vivo study or a clinical human study. And completion of this workflow really requires the software to be both structured and flexible. So the flexibility is essential while this method is being iterated on in this first stage. And then once that method gets established and validated in some instances, then the processes become more structured and the method is standardized to capture and report on those preclinical findings. So for the live demo here, I'll be touching on some key aspects of each one of these different activities, and the process was within them. So for method development, looking at how one might capture some of those methods and gain some insights from them through study planning and establishment of those SOPs, and then through the sample testing flow. So sample management, prep, data analysis, and then finally witness and reviewing of that data collection. All right, so let's get started on the platform. Oop, there we go. All right, so as I had stated, right, one of the first deliverable here is method development. And method development can take a lot of different shapes and forms. I have got a really brief example of here, but these can extend from things like identifying key reagents, calibration curves, QC samples, establishing any number of other parameters. So here, this is just a method, an example of some result capture, establishing some activity parameters for an ELISA assay for our drug candidate. So here at the top, I have mentioned my drug candidate here, this antibody, and have used a plate template to plan out my experiment, where I've got a few different dilution series here, and then I'm going to be capturing some data on those dilution series. And these are all structured in a stepwise fashion. So as I come through, even though this is more flexible in nature, I still have some structure to how I'm performing this assay. So here, this is already partially completed, obviously. So I've already completed or entered the data for two different data sets. So the 1 to 2 dilution and a 1 to 5 dilution. And after entering the data, I used benchling to try to fit a EC50 curve to those data sets. So for the 1 to 2 dilution, it was too flat and it couldn't calculate a curve. For the 1 to 5, though, we got a kind of the beginning of a curve, so we can see we're up here right at the top. So this is a snapshot here from our Insights platform. And let's look a little bit at that last data set. So we want to capture that 1 to 10 dilution. And in a low throughput fashion, typically what we'll do is upload that directly from either our instrument or maybe from a parsed source, where I'll just drag it in, that data set off of my desktop. Eventually then will map the column headers within my CSV to the structured data capture within that table there. And you'll see these all line up and perform a quick validation step. and then import the data. So now that they've been imported here, we'll see that we've captured our different protein concentrations and activities and dilution factors, but I have to actually submit it. So this is one aspect eventually in which we're controlling for submissions. So unlike some free-form, non-structured tables that you may also work with. This one actually, after submission, the only way I can remove any of these rows is actually through archival. So really maintaining that traceability of data capture and entry, even if I needed to change things. So this is kind of a brief example of kind of maybe how I would go about fine-tuning my assay with different dilution curves. But ultimately, what we're looking to do here is to gather all of our best practice parameters and then capture those within a structured fashion. So the way in which we'll do this after our method development is complete is by actually registering those parameters within the database. And this serves a couple purposes. One, it serves to create a single source of truth for your for these different parameters, but also allows you to resurface these later down the road so that you can use them for comparative analyses. So here, where it would be something of the sort of like ELISA method, and then I could link it to that drug candidate that I was just researching, maybe a specific activity and others. But moreover, I can actually link my SOP, that standard method SOP. So depending on your need, you may have an electronic QMS where you're actually publishing and managing the doc control there. And this can be a hyperlink to your EQMS, or this could be created within Benchling, or even just a Word document. So here is kind of would be an example of just maybe like a Word doc version where I could import that and directly associate it with my method. All right. So that's one way that we would look to capture those criteria, um, but and then take them forward into a actual bioanalytical study on some ongoing in vivo work. Before I do that, though, I want to just briefly look at this data, right? So we just captured this set of raw data, and I had talked about establishing this curve. So I'll do that here through use of an actual dashboard that will automatically graph it. So you'll see the live view of that 1 to 5 dilution factor. And if I want to view that 1 to 10, I can simply just change my parameter here, and we'll update my view. And you'll see here now that I've got a wider range of dilutions with my 1 to 10 dilution series. I've got a nice curve and a calculated EC50 here that I can then transfer over into my experimental record before submitting. All right, so moving beyond this, as I alluded, really the next step of our process here is to establish a study plan. And we'll do that in Benchling through use of a templated study plan. So where this is mostly completed here, where we've identified our objective and we've utilized at mention functionality within Benchling to collaborate on creation of this study plan. So where I can make callouts to our formulation team to make suggestions on which lots we should use for our testing, or the analytical team to actually be the primary author of this, coming through some key deliverables in creating this document, and then finally quality actually reviewing this for reviewing the content. So to achieve those different things, we've surfaced a lot of information right from this one workspace, where we have our purified protein that our formulation team has entered here, and it's automatically looked up. Has been a bunch of key information about that. That's all stored within that protein, as you can see here. So it's surfacing this information within this entity, this protein lot rather, right here within the study plan. And then we're defining the key aspects of that study, right? So not only naming it, but what types of samples. So this would be, say, in the context of maybe an in vivo mouse experiment or study that we're receiving samples off of. We have a few different matrix types, some different time points, and just overall characterizing these. But you also notice here we have our methods that we've established through the method development process linked here within our study. So we know wherever we're at within the study that we can surface these methods, and not only just the piece that we have registered, but also the direct link to that method. So that's also would be surfaced right here, so that we're able to visualize this as we move through the process. And just quickly verify that and commit that to our record. All right, so these are some of the structured ways that we capture this information. Clearly, there's be a lot more detailed information within this study plan around actual actions that may be taken, or more granular details on reporting requirements. You get a good sense of here some ways in which benchling is utilized to create structure and capture within capturing both the SOPs, but also in the context of the greater study plan itself. All right. So the next step of the process, really, so after our study has been characterized, or completed, the study entry has been completed, it has to be submitted for review. So we're going to do that here through this review process. So this is a part of Benchling's 21 CFR Part 11 compliance, and can be leveraged for GXP or GMP type operations. But this is the element here where I'm showing where we're sending this out for review. We can then select a predetermined list of auditors, leaving a comment. Study plan is complete. We'll create that review and lock our study plan pending review. So we'll see here at the top, it's now needing review and pending, and we can no longer make edits. So that would be kind of our workflow for the author. If I think about the reviewer, they're going to receive a automatic notification that this is ready for their review, and be directed to this study plan where they'll come into the review tab and be tasked with a specific reviewing this study plan for a specific set of criteria that you've determined. It's like capturing a lot of information, that we've registered those key elements into the database and documented those metrics. And then if they've passed, then approving that study plan for further to move forward. Okay. All right. So once that's all been performed, really the scientific aspect or the testing portion of the study is going to kick in, and we'll manage that through use of the workflows application within Benchling. So to first kick that workflow off, the analytical team is going to create a new workflow from, say, sample receipt, where then I'm able to assign a receiver for this study. I'll assign it to myself here, referencing the study plan that we had just created, as well as potentially a delivery date from our in vivo team. We'll set that out into the future. And the actual manifest. So whether this is coming from an external partner or maybe internally, manifest could take the form of, say, an Excel document that could be attached here. So if I attach that manifest, I'll be able to import that and kick off the first task of our study, which is receiving the samples themselves. All right. So you see here that this is now been represented as a task or an input, and also the output of deliverable of that, which is going to be the actual sample barcodes. Before that, though, this is the view that I would take. So as the fact that I've just been assigned this, I'll now, me personally as the assignee, is going to receive an email notifying me that this this task is ready and been created for me. So I can then come to the dashboard, and if I'm curious about what samples I have to receive, I have the ability to create some saved searches or saved views where I can say, hey, what are my sample receipt tasks that are assigned to me here? And I'll see two here. So this is the one we just created at the bottom, and one that was created prior, which is currently in progress, all of which have the full context of that information we created. So I'll jump ahead a little bit and jump into this one that we had just seen. So we can now think about how we would translate that manifest into uh, something we can understand within the database. All right. So after executing this task, well, I'll show a little bit more about executing in a moment. But after executing this, we have a populated template here that is structured in nature, bringing me through step by step, and can be configured to be completely locked down, so I can't edit any of the text and only check boxes and interact with certain areas of the platform. That all that sample context and manifest has been brought forward to this record, where then I am able to download that manifest directly, and then insert that directly into my database here, so you can see essentially what it looks like. And I've used that and uploaded that manifest within this structured table that's gonna submit this to the database, to the registry. And at this point we've captured some sample specific information, like what time point. But now that the sample has been registered, our new registered sample is completely washed of any identifying information, so blinding any of our future efforts. So from sample prep or the sample testing, those analysts won't have this information to reference. So after registering, then I'll have to transfer that to physical inventory. And that's also all done within the stepwise execution uh, template. I've registered and then moved on to transferring. You'll see here I've transferred them into a sample box. I click into that sample box here. You'll see I've populated it with 90 of these samples that were registered, and um, contained within that box for transfer to the next team. All right. So lastly, I'm going to have my task to receive these samples, but then I also have my output here. So I've added these barcodes directly to my output and automatically associated them with the task above. We'll look a little bit about this a little bit later. And then once this output is recorded, it has a unique output identifier for each one of these barcodes and associated tasks. All right. So lastly, so now that my work is complete, I will completely indicate that the sample receipt has been completed and submit that. And now I'm no longer able to change the status of this task now that it's been completed. So for the next step of the process, we're going to be, now that we've received the samples, we're going to actually send them for preparation. So to do so, I'm able to leverage this same workspace where I have my outputs, and I can directly link those to a series of inputs for the sample prep flow, where we have our sample prep. This is automatically mapped for me, so it's pulled in from my output directly into my new set of tasks or inputs. And I'm able to, we'll say, assign those to a specific uh, folder, and then create those sample prep tasks. Now we'll see once we've created these, they're going to come in with not only just the barcode information, but also some additional context for our next analyst that's going to be actually doing this. So we have our barcode information that we input as part of the output, but also carried forward are the study ID, the sample ID, sample type that was contained within the manifest, but also the method here. So we have some additional test methods that are also going to follow us through here. So when then they go to execute on this, they'll be able to have that context. And I'll show you how that's executed on now. So for our sample prep, this may be a different individual that's performing this. So they would come into the lab and wonder what they have on the docket, and they can look at just what are my sample prep tasks through another tailored view that's unique to their user. Where here now I'm stepping in the shoes of Lauren, where she has completed the sample prep for a prior sample receipt. So again, she has all this information from the sample to the sample type, and can execute on these, where she'll select all of them and then be able to execute these. Well, because these have already been executed, let's jump into some that haven't been. Let's just pull up, we'll look at all those biopsy tasks. So now we'll look at all those biopsies just to show you how easy it is to execute on them. And then also they're executed in a structured template, an execution template, where I can batch together any number of these samples and execute them into a new template that is going to then batch together and pull in all of these different samples into one work stream. So I have here all of these biopsy samples from these studies right at the top to be able to reference and complete my sample prep. Right. So as the analyst that's preparing the sample, we'll see what Lauren did here. So within this template here, has the ability to drop in her SOP. So this is particularly useful if you have an electronic QMS, where you can download that most up-to-date current version and drop that directly into this execution template to record what method you used. So she's using a tissue prep, and then utilizing the same structure where she's registering those sample preps after they've been created and transferring them to inventory. Now she can pass off to the testing team. All right. So the last stop we're going to make is on that testing side, where we've executed on a few of these requested assays and captured some information via our instrument. So for our sample ELISA, so our testing analyst here, so we'll have Lauren. She's gonna do that again. She has that full rich sample context pulled to the top, so that when she needs to know what method to perform, she can click back into her method here and have easy access straight to that testing document, that established validated method. All right. So once she downloaded that method, she would then drop that into her document, establishing which method was performed, and be able to have easy reference as she's performing the assay. So one aspect of this, because her workload may be large, is the ability to automate some of these data capture processes. So she's utilizing the laboratory automation aspect of Benchling, where she's able to upload a raw data file or even post these via the API automatically directly from her plate reader to measure these results. So in that sense, we're going to just drop them in, but she'll drop in her lab automation, drop that not in the right place. drop those into her run here, and then let Benchling do the rest. So where she'll be processing that and capturing and automatically assigning those results directly to the samples that were on the plate readout. So here, see here, we've captured all of this rich information straight from the plate reader. All right. And then lastly, just similarly, she'll complete the output. And all of the, hopefully you've been able to see here that throughout the workflow, at any step of the process, each of the analysts that were assigned had all of the information they needed to perform the experiment. So the full context of the study, the approved methods for that study, as well as the samples and barcodes that are associated with those, and being able to track back the exact inventory locations where these exist within their lab space. All right, so with that, I will jump back over and hand off back to Prem.
Thank you, Tom, for that demonstration. So just before we head into the Q&A session, just I wanted to touch upon some of our customers. So Benchling is excited to really help development teams. We're proud to work with over 600 industry customers across R&D. Let's see. And just to summarize, Benchlink's solution for bioanalytical development helps improve data integrity by standardizing data management, facilitates collaboration by providing access controls, improves access to all your bioanalytical data at both the experimental level as well as the operational level, and provides a multitude of built-in tools for ensuring compliance. With that, we will head into the Q&A session. Please, if you haven't done so already, please type in your questions. On the right side, there is a Q&A tab. Please go ahead and type your questions and we will go through. I see a few questions here already, so we'll start answering them in the order that I see here. And yeah, feel free to keep adding to the discussion as we go along. So the first question we have is from Cheryl. The question reads: are we able to set up these processes ourselves, or is someone from Benchling able to set this up for us? So maybe Tom, like, can you answer this question? Like, maybe talk a little bit about how much of the configuration can be done by customers versus where they might need like Benchling services support.
Mhm. Yeah, absolutely. So, our customer success team and customer experience teams will help you to establish some of the best practices and train you on things. But the everything we showed for today that I had just shown was all actually configured through a point and click interface. So all of those tasks and all of those fields really you can be edited within a matter of seconds, really going in using drop downs and making different linkages. So certainly all of these tend to be tailored towards the exact needs that you would have.
Great. Thank you, Tom. The next question. So next question we have from Manju. It reads: is this platform compatible with Watson Limbs and similar software? I think, yeah. So I think this might be a good opportunity, Tom, to comment on our developer platform and some of the APIs we allow for integration.
Yep, absolutely.
Go ahead.
So all of the actions within the UI that I was walking through, so interaction with the tasks and the outputs and creating all of those, all have a full set of REST API endpoints. So if you want to create new tasks and things like that, can all be done programmatically. And regardless of what database you're using, if you're using a LIMS another LIMS software, those connections can easily be made within Benchling. Also, just mention too that it can be particularly useful. We have a developed events platform as well that is can kick off things or give notifications through the Amazon Web Service Event Bridge.
Right. So just to reiterate here, right? Like, I think so. Benchling is essentially, it can be like a LIMS replacement in itself. So if you want like a complete all-in-one solution with ELN and LIMS and LES capabilities all in one, Benchling can be that unified solution. But in case for whatever reason you do choose to keep, let's say like Watson LIMS in this case, or some other softwares because of other internal considerations, then we do provide the tools and the developer platform to allow for integration with these external systems. On to the next question. What is the advantage as compared to ELNs? So maybe like Tom, I think you can comment here, and then I can chime in as well.
Yeah,
Yeah. So
absolutely.
how
So
does our benching notebook stand out from other ELNs?
yeah. So you can think of the Benchling notebook, right? So or think of it actually instead of it just templates in general. So templates within Benchling can be configured to reflect something like you would be familiar with like an ELN, very modifiable, very word processor oriented, but then also through uh, a number of features like locking down certain elements within those tables or locking it all together and making it fill in only. You can really tune it and turn the knob to a lab execution use case.
Yeah, and just to add on to that, right? I think the other major differentiator for Benchling Alien is really that it's actually built on this unified platform, which means you enter data once and it shows up across all the other applications. So maybe you might register a sample in your notebook, and then you can look at the same sample and related data in the registry, for example, or the inventory, and you can and or maybe you can click on the sample ID in your notebook, and it takes you to the actual inventory information. So just that interconnectedness of all the different applications, I think, is the other major differentiating factor of the benchling ELN compared to other ELNs. Uh, Next question. Okay. So the next question we have from Manju is user access controlled? So maybe I can go here, and Tom, feel free to chip in as well. So benchling offers a pretty in-depth uh, access policy and like permissions system. So what this means is that you can really control access by user roles and personas at a very granular level. So we have in addition to basic access such as read, write. We have, I think, author only edit access. We have a pretty comprehensive set of tools here. Do you want to add anything more there, Tom?
Yeah. So that's definitely a good summary of the user policies themselves that are associated with each user, depending on where they sit within the organization. But they also are governed by project permissions.
Mhm.
So this is particularly useful for if you need a clear division between, say, a more clinical oriented team and a more R&D oriented team. So any data that are collected in the project folders are governed by those permissions. So even if they share a
Mhm.
sample ID, I can only see them based on what permissions, what projects I have visibility to.
Mhm. Good. Thank you, Tom. The next question we have from Mark. It says, how easily can sample study data be corrected, and how are these corrections tracked audit trail within the system? Tom, you want to take this?
Yeah. So I had shown you a structured table early on that where it captured some analytical data off of that ELISA. So once you hit the submit button on any of these tables, it can't be permanently deleted. So when you hit submit, that's part of the permanent audit trail within Benchling. And if you needed to make a correction, you can archive, right click on the row and archive it, and then be given a few different options around reasoning and as to why that data needed to be archived.
Mhm. Yep. Yeah, so yeah, exactly. So basically, everything on benchling is like tracked, right? Audit trail, like even though, yeah. So that in addition to the archival system, we have one more question. Okay, the next question we have is, what type of analytical instruments can we integrate with? I think maybe I can just start off here. So I think so. Benchling supports a few, like I said. I think we have an open developer platform, so all our API documentation is public, and we support like REST APIs and event APIs. Like those are the two main ways of integrating with Benchling. So what that, but the REST APIs is very, it's a very common standard for interfacing between data systems. So because of that, we can integrate with a wide range of analytical instruments, and like right from like plate readers to freezers, like to smart, to smart freezers and analytical instruments. So I don't think there is any limit to which particular ones we can or cannot. I think, yeah. So maybe Tom, if you want to add anything there.
No, yeah, that's exactly right. So, the integration is a question that means different things to different people, and we can support the complete range, whether it's just data mapping or all the way through full programmatic data capture, much more hands-off type of approach.
Right, exactly. And the other thing also to highlight there is, so the integration can be done at the customer end as well, because our, like I said, our API documentation is public, so you can do some of these integrations yourself, or if you need like benchmark support, we also offer like comprehensive professional services to help you here. The next question reads, can I integrate with third party analytical tools such as Tableau, Jump, or Spotfire directly? Tom?
Yeah, so all of those business intelligence type tools that you may be familiar with are easily connected through the data warehouse. So I showed a snippet there with our own internal insights analytical tool, but that's all querying a Postgres warehouse. So those credentials can be made available to directly connect to Jump. So you can have a read-only view of the data within the warehouse.
Got it. Yeah. Thank you, Tom. Next question we have from Nicole. When adding samples to a workflow, are there options to gather samples from the main database for all samples received in Benchling? Mm, let's see. Yeah, so I think maybe if I understand this question correctly, it sounds like maybe there's a pool of samples that somebody received, and then you want to maybe like analyze all of them at once, or maybe a sub portion of those samples. And I think benching has what we call work lists. Like maybe Tom, you can talk about like how work lists can be used to process samples in batches.
Yep. Yeah. Yeah. So you can certainly select and create a work list out of anything, whether it's containers or registered samples within Benchling, and those can be used across the platform in any structured location that exists within it. And specifically to workflows, you can. Yeah, there's full searchability and filterability of those samples that are in that workflow, the row of those task rows, and you can batch together samples that may span multiple different workflows or samples. So if I'm an analyst, I can pull things and batch together to fill my assay, the assay plate that I'm running per se.
Mhm, mhm. Cool. Thank you, Tom. We have a question from Natalia. So the question reads, who has access to the data? Only our company, or is it open source? So I think I want to be very clear here. Like on Benchling, if you are a customer of Benchling, the data on Benchling belongs to you, right? Like nobody, like even we don't have access to your data unless you explicitly give us like a Benchling support access. We have customers where it's completely like Benchling is completely blinded to what you do on the platform and how you use the data. So yeah, so the data on our platform is completely yours. It's 100%. Yeah, it's not open source or anything. We have a question from Matthew. Are we able to track, capture sample movement, location, freeze thaw cycles? Tom, do you want to comment on this?
Yeah, absolutely. So I showed a little snippet of what a box looked like inside of a box, but any of those either clicking on the vials within the grid or otherwise can be selected and transferred to other boxes. It's a simple move action. And in terms of capturing freeze thaw cycles, it's typically done through linkages within the database, right? So imagine a cell line, which it serve as a parent descriptive object, and then there's a child that relates to it, and that child could be your cell bank, which could have its own set of specific information or tags like what the freeze thaw cycle is. So create these relationships to be able to track those sorts of attributes.
Good. Good. Thank you, Tom.
Mhm.
The next question: can you talk more about role-based permissions in benchling? I think we already discussed that at length. I think somebody else had asked the same question, so I think we can skip over this question. Will this require additional resources in the lab? I think like this question is fairly open-ended, but I think if I understand, maybe like Tom, we can comment on like what aspects of benchling requires like special, like maybe an admin to manage like the benchling software. Yeah, so if you can just talk of like some admin roles versus like user roles, I think that maybe that can answer this
Yeah.
question.
Yeah, so the benchling exists as a configurable platform versus something you would think of as requiring some sort of internal custom engineering on benchling side. So with that in mind, the configuration is point and click. So, but that is limited to just the admins within the organization. So they would manage and curate all of the different configurations that really power the platform. Whereas a standard user would only be able to interact with them.
Right, exactly. And I think just to add on to that, we also have like a tenant admin console. So every deployment of benchling would have an admin who have access to that special dashboard where they can add users, delete users within the organization. So they have this sort of like the super admin within their organization. So I think that's the only extent of extra resources you might need. Yeah, if you want to just get a greater control over your own like data management practices internally.
Yeah. I would just add too, right? And if you're thinking about it in terms of if you need an FTE in your organization that is has technical skills like IT type technical skills, it's completely not required because we'd have a lot of smaller customers that just have a super user, which is essentially a scientist that can be trained to how to operate the system and how to manage the configuration.
Yeah, great point. Yeah, thank you, Tom. Uh, On to the next question. How do I manage the visibility of my data, especially outside of my team? I think this is worth answering, like because we discussed about access policies and permissions. Maybe we can layer on like how some of that can be used to manage, let's say, like a CRO collaboration, right? And we mentioned that in the presentation as well. Like how let's say that be set up. Can you talk through an example, Tom?
Yeah. So there's a couple different ways. I touched a little bit on managing visibility and accessibility through permissioning structures on the projects themselves. So a project can be defined as anything that, however you delineate your work within your company, either at the team level or maybe campaigns or things of that nature. So you can provide access to those. So a lot of our customers will actually have a seat for CROs that they can access directly and get into benchling, but in a restricted fashion, just to individual projects. But if you need just individual access, any one of those execution templates or entries can be created as a shareable link to share outside of your company or organization.
Great. Thank you. We have a question from Cheryl. Would it also be able to connect with FreezerWorks? So from my understanding, like FreezerWorks is basically an inventory management software like FreezerWorks, Freezer Pro. They are all specialized inventory solutions. So yes, we can integrate. I think we have some instances where we have been able to do that when customers want to keep their inventory solution. Yes, we can integrate with these other softwares. The next question says reads: can you generate data tables, graphs, reports within benchling? I think I can comment briefly, and then you can add to that, Tom. So I think there are two ways, right? So the way to generate like graphs is obviously through our like insights platform where you can query your data sets across like entries, across projects. Essentially, any data that's in benchling you can query and visualize using insights. So that's the way we think of it, as a way to aggregate data across like multiple data sets, right? And so you can aggregate, you can visualize, and from there on you can export if you want to run like more advanced analysis elsewhere. And in terms of like like tables and reports, I think those can be done directly from within entries, whether it's like notebook entry or whether it's like search. If you want to search for a bunch of like different samples, you can export into Excel. So that's the other way, like where you can like export data into tables, and reports I think can happen directly from each individual apps, say whether it's notebook or registry. I think you can export into PDF. Anything else you want to add there, Tom?
Yeah,
Just
yeah. So you can also set up designated templates that will serve as some sort of a certificate of analysis or something of that sort, where you can look up information from the database and aggregate it in tabular fashion within a template. That's we certainly have folks that are doing things of that nature.
Cool. I know we are over time, but there's still a few more questions. So let's just run through all those, just a few more questions, and then we'll just go over slightly. So next question from Kevin: is there a way to track samples that leave benchling if they are shipped offsite or discarded? Tom, you want to take
Yeah,
that?
yeah. So in terms of shipping, right? So if you have a deliverable that you actually want to ship, a common use case is to actually input, say, tracking information into associated with that shipment, so you can have a record of that aspect, that action. And then if you're removing things from the database, you can also utilize the archiving functionality when you're talking, say, about containers, and you can archive because things are expended or shipped, or you can indicate those sorts of reasons for archival, and instead of just made an error. Yeah.
Got it. Yep. Thank you, Tom.
Mhm.
Just a couple more questions here. Can you perform the data analysis within benchling similar to Watson limbs, regression analysis, for example? So I think like now, so with that insets product, we do allow some of these analysis, like regression, like maybe Tom, you can comment on some of the specific types of curve fitting we can do now.
Yeah,
um,
yeah. So the curve fitting right now, this is a very active area of development for us, and I had shown you a logarithmic regression for EC50 calculations. You can do things like linear regression, and there's also heat map. You can create some heat maps based on the data that you have, and so that would be current state, and this kind of an area of active expansion, though. Yeah, so it depends on what you're looking for.
yeah, exactly. We are building many more like scientific analytics tools on the benching platform, but we have some of the core functionalities that scientists might need for bioanalytical. Um, the next question we have is, can you lock sample results and flag samples that have been tested? Tom, you want to take this?
Yeah, yeah. So you certainly can do through the use of looking up of key attributes, create conditional statements within the data capture to pass or fail things based on those QC parameters. So if you have a specific metric, then it can create a pass fail criteria, and it's not editable by the user. So it's really just about what the data are, and how they match up to standard.
Got it. Thank you, Tom. I think, yeah. So those were all the questions we had. So thank you so much for engaging with us, attending this presentation, and asking all these great questions. We just have one last poll for you before you sign off. Please, please vote on this poll. The poll asks about how useful you found today's presentation, with one being not useful, two being somewhat useful, three useful, four somewhat useful, and five being extremely useful. Um, we'll just keep the poll open for a few seconds. Uh, all right. Thank you so much, everybody, for joining us this morning. Have a great rest of your day. Thank you.